Okay, hello everyone. Welcome to today's special seminar. So I would like to begin by acknowledging the Wiradjuri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining us from today. I pay my respects to their elders past, present, and emerging, and to the Aboriginal elders of other communities. My name is Sepide. I am a postdoctoral research fellow at CCM MP from Melbourne University Note, and I will be chairing today's seminar. So before we start, I just want to go through some housekeeping rules. Just to let you know that the seminar will be recorded and it will be available in CCMMP website later. So please stay muted throughout the presentation. So there will be a Q&A session at the end of the seminar, which you can unmute yourself and ask your questions. You can also type your questions in the chat during the seminar as you think of it. It is my pleasure to introduce today's speaker, Professor Ariane Briegel. So she obtained a master's in biology from Ludwig Maximilian University and PhD from Technical University at Munich, Germany. She then moved to California as a postdoc and later research associate at the California Institute of Technology. She's currently a professor at Leiden University in Netherlands, co-director of the Netherlands Center for Electron Nanoscopy, also at Leiden University, and holds a guest appointment of medical delta professor at Delft University of Technology, Netherlands. Ariane is currently an advisory board member at the Cryo-EM Center Nuremberg, the Cryo-EM Center Pasteur Institute in Paris, for Cryo-ET 101, University of Otto, representative for Instruct Eric Netherlands, a NWO Research Community Committee member for Advanced Methods, and mentor of Dynamon's mentoring program, University of Hamburg, Germany. She has over 20 years of experience in studying bacterial by cryo-electron tomography, and her laboratory uses Cryo-ET to study the structure and function of the molecular complexes involved in the bacterial cell's ability to sense and respond to their environments in their native states. And today she's going to talk about how cryo-electron tomography contributes to our understanding of bacterial interactions with their environment, and actually how do they interact with phages, how they can evade toxins and predators, and how they can adapt to their and thrive in changing environments. So basically, Cryo-ET is a research tool that is used to gain insight into the structure and function of the molecular complexes involved in these kind of behaviors. And these techniques allows the direct study of microbes in their native states at resolution, which is capable of visualizing individual proteins. So she's research provides insight into chemotactic behavior of bacteria, which allows motile cells to detect changes in nutrient concentration and to navigate towards preferential environments. She uses this detailed knowledge for some practical application as well, such as the design of biosensors to detect human diseases. Ariane is also investigating a specific adaptation of chemotaxis systems of human pathogens that use this system for infectivity. So I guess I'm not going to just without further ado, I'll just hand it over to you, and please, whenever you are ready. So.
Good morning, everyone, or good afternoon for you. It's a pleasure to virtually visit your institute, and I'm happy to tell you a bit about what my lab does. And so I think you see on the first slide here already, I do like sci art that I think really helps to convey some more difficult scientific questions. So on the slide here, you can see like really the core interests of my lab, like how do bacteria sense their environment? How do they navigate it? How do they interact with each other, either by combating each other or by forming communities like biofilms? How they can infect a host, or how they interact with viruses that infect them. We do a lot of, there's a lot of questions in the lab. So I'll show you like a selection of the topics we work with. Oops. Okay. So the reason I came to the Netherlands is I'm tied to very large instruments, very large microscopes. And here you can see me standing in front of one of our microscopes in our facility at NeCEN. This is one of our Titans. So we really depend on these big instruments to look at our very small samples. Now, I spare you the technical details on how we get these images. I'm happy to talk about this later. You also have an expert on this, I see here in this talk today. So Debnath also can answer all the questions if you have more. Hi, Debnath. So I just want to show you what kind we actually can get with this technology. And so here you can see a very interesting bacterial cell called Bacterial Vibrio. So these are cells that can prey on other bacteria. So they swim through the environment. When they encounter a host, they slip into the cell envelope of these host cells and then digest them from the inside. Then they multiply and they burst out and find new hosts. So here, just to show that the power of this technique, we can see all these different components of individual cells at macromolecular resolution and in 3 dimensions. So you can see the cell envelope structure. You can see in the tan color, it's the chromosome of the cell. In yellow, the ribosomes, and we have cell attachment structures like pili that the cells use to attach to a surface, or flagella that they can use to swim around, or the chemoreceptor arrays in dark green, secretion systems, and so on and so forth. So it's very powerful to look at individual cells and study their molecular structures inside. Now, as Sepide already mentioned, one of the core behaviors or core behaviors of bacteria that I'm interested in is their ability to chemotax. So that means the cells can sense their environments like sugars and amino acids, or also toxins, and then they can steer their motility toward or away from such substances. So here is a movie I took a long time ago. I think maybe we were still in the same lab when I did this. So I put a sugar source at the bottom of the slide, and you can see E. coli cells can sense the sugar and can actively move toward the sugar source. Now, how do bacteria do this? So in E. coli, the cells have about 8 to 10 flagella distributed all over the cell body. And when all the flagella motors, you can see here a structure on the left, when they all rotate in the counterclockwise direction, all the flagella bundle up behind the cell in one large super bundle, and they push the cells forward in a very smooth motion. And that's what you can see in the video in the center when they zoom around. So this is several flagella bundled together that propel the cells forward. Now, occasionally, one or more of these flagella rotate in the clockwise direction. So this bundle dissolves, the cells tumble around until all the flagella return to the counterclockwise rotation again. And so by tuning the duration and frequency of these run and tumble phases, the cells can move up gradients of attractants or move away from toxins. Now, how do the cells really control where they go? And so here we have a schematic of how this works. So I already introduced the flagella motor on the right here, but on the left, this is the sensing part. So these are the chemoreceptor arrays in these cells that allow them to sense the sugar or toxin sources. And in E. coli, it's deceptively simple. So there's only 11 characterized core components that provide the sensory system. When you compare it to, for example, eukaryotic signaling pathways, where you have often hundreds of components helping the sensory pathway. So this is really a really nice model system because it's very simplified. So how is it composed? So we have chemoreceptors that insert into the membrane here in E. coli. So there are thousands of these receptors. They cluster together with 2 additional proteins. One is a linking protein called KW that helps the arrays cluster together. And then we have the enzyme of the system. It's called Ka. So these 3 components, the receptors, KW and Ka, cluster together, form these large complexes at the cell poles. And then when something attractants or repellents bind to these receptors, the receptors control the activity of the kinase, which in turn transfers a phosphoryl group to a shuttling protein that then goes to the flagella motor and controls the spinning direction. Now, chemotaxis, it has been known for a long time, like in the mid of the 1950s, already people started really investigating the system. And so we know all these core components. And we also have crystal structures for all of them. So here is one of the E. coli receptors from the crystal structure. They form these ice cone-shaped trimers of receptor dimers. And on the right, you can see a cartoon of these receptors. So we have the periplasmic domain where attractants and repellents can bind. And we have a transmembrane region, and then a very long coiled tail that sticks into the cell. So the active enzyme of the system, again, this is called Ca. So you don't have to remember all the parts here, but it's two parts of this. So we have a structural core. This enzyme forms homodimers. So we have a dimerization domain, and then the P5, the receptor binding domain, and the kinase domain. These three domains form a structural core of the protein. And then there are two additional domains that are attached via flexible linkers to the rest of the protein. So one contains the substrate histidine, and the P2 domain, that's where the shuttling protein is bound to receive the phosphoryl group when it's activated. So we have a structural core and then two domains on flexible linkers. And then last but not least, I mentioned this linking protein that is structurally very similar to the receptor binding domain of the histidine kinase. So you see here a co-crystal structure of the P5 domain of Ca and the linking protein. So we had all these structures, but when I started working on this, we didn't know how these arrays actually form inside the cell. And one of the complications here is that these large arrays are just too big for a structural technique like crystallography. So tomography really currently is the only high resolution method that we can use to study these arrays when they're assembled. And this assembly is important for the proper function of these arrays. So for their cooperativity, for example, the arrays have to form. So how do they look when we look at in tomograms? So again, on the left, you can see the cartoon. We have the receptors, the linking protein, and the histidine kinase. And then next to it here is a slice through a tomogram of an E. coli cell. And you can see I highlighted here the components with color, so it's easier to see. So in red, we have the chemoreceptors. Here, right underneath this yellow arrow, we have these, you see these little dots. This is the periplasmic domains. Then we have here, this is the inner membrane of the cell. And then the long tail, coiled tails of the receptors. And then where this blue line here is where the kinase and the linking proteins bind the receptors. So now, fortunately, we work with three-dimensional data, so we can look at the arrays in top view. So we rotate this volume 90 degrees, and we look from the outside of the cell toward the inside of the cell. We go through these arrays. So this loops around. So here we come from the outside. Here you can see the trimers of receptor dimers, and then underneath these densities here are the kinase domains. So I think what I want you to remember here, this is highly ordered. This is a near crystalline lattice inside the cells. So it's highly ordered, both from the receptors as well as in the kinase. So this is E. coli. Now our resolution is not as high as what you would get from crystallography, or if you would do single particle cryo-EM. But our resolution here from inside of cells is about 15 angstrom, which is good enough to dock the crystal structures we already have and really build these arrays into the densities that we get. So this is what you can see on the right. So the gray densities is from our tomograms, and then we dock the receptors in red in there, and the kinase or the enzyme in blue, and the linking protein in green. So already a number of years ago now, we could solve really the architecture of these arrays in E. coli. So here is our working model on how signal transduction works on a structural level based on our data. And so here, this is the smallest functional unit of signal transduction in chemotaxis in bacteria. So we have 2 receptor trimers of dimers, 1 kinase dimer, and 2 linking proteins in green. So here, the receptors are in a closed conformation, and the flexible domains of the enzyme are bound underneath. When the receptors get activated, they open up, and in turn that triggers a release of these flexible domains of the kinase, which allows the shuttling protein to bind and the phosphoryl group to be transferred. Now here is a cartoon of the situation in E. coli. I mentioned these are extended arrays. So these enzymes and the linking proteins form these rings underneath the receptors and form these extended lattices. When we first started to describe these architectures, we had a lot of reviewers who were a bit doubtful that this is really as universal as we claimed. So I followed this up with a study where I investigated chemoreceptor arrays all over the phylogenetic tree. So you can see here in this phylogenetic tree, all the strains marked here with a blue asterisk is where we collected data and looked at the chemoreceptor arrays and even the chemotactic archaea. So in all of them, the receptors were packed in this universal architecture with this characteristic arrangement. So this is the state when I moved to Leiden. Now, when I came to Leiden, I decided to revisit this question, and we decided to look into chemoreceptor arrays in pathogenic bacteria, and we selected 2 different ones. One is Vibrio cholera, which I won't show you today, and this is the other one. So this is Treponema. So Treponema denticola, to be more specific. So Treponema is part of the spirochete family. So spirochetes are quite nasty pathogens for diseases, for example, syphilis, Lyme's disease. And so this one is also a pathogen, but maybe not as scary. So it's causes periodontal disease. So we decided to study the chemotaxis system in these bacteria. As here we know that chemotaxis is actually the first step of host invasion. So it's a potential target for possible treatments in case of antibiotic failure. So we were very interested in studying this. So we first visited the architecture of these arrays, and interestingly, we found that while the receptors indeed were also packed in this hexagonal fashion, like in E. coli, we could find that the underneath the lattice of the kinase had a different arrangement than in E. coli. So again, the situation in E. coli is on the top. The kinase is here labeled in blue, underneath the receptors labeled in gray. So this is the situation I showed you before, on the receptor level and then on the kinase level. And when we compare this to Treponema, we can see that there is also this hexagonal arrangement, but the kinase is arranged in these linear rows. So at the time, when we done our first study, we could show that actually this arrangement is likely due to the different cell morphology. So these treponemas are very, very slender, thin, curvy rods. So the chemoreceptor has to have a much higher curvature. This arrangement of the kinase actually allows the arrays to accommodate this curvature. But more recently, we continue to study this, and I hope you can see this. So here is the E. coli situation again. The white asterisks mark the individual receptors of one trimer of dimer. And so here we have the situation in treponema. And for the first time, we see this extra density in the center of these rings. So we're very puzzled by this because we had looked at many, many different species. We've never seen anything like this before. So we wondered what this might be. Now, when we look at the genome of treponema, we find that there are actually two linking proteins encoded in the genome. One is a classical KW, a classical linking protein like in E. coli. And there's a second variant of KW, which also has one of these KW domains, but it has another domain attached to it, and that's called a KR homolog. So KR is a methyltransferase that's also used in chemotaxis to adapt to their adapt the receptors to the current situation. So there is a methyltransferase and methylesterase that together control the methylation state of the receptors, and that is also controlled by the kinase. So it basically tunes down the activity of the kinase when it was highly activated if the conditions stay the same. So it's kind of a memory system for the receptor or for these chemosensory systems. However, the methylation sites of the receptors are further up towards the membrane. So this KR homolog is structurally not able to ever reach these positions on the receptor. So it has to have a different function. So initially we found that these KR homologs, they in the crystals they do form dimers. So this was our model, that actually these KR homologs dimerized underneath the arrays and support the stability of the arrays. And indeed, if we remove these domains, these arrays become smaller and structurally less stable. Now here, we did some molecular dynamics simulation of these KR-like domains. This was done by Keith Cassidy, our collaborator. At the time, he was at the University of Oxford when he did this for us. So here we have this layer of the linking protein and the kinase that you remember now from E. coli, and then these additional domains of KR, they dimerize underneath this, so the receptors would be above here. And now if we turn it around and look from the top, again, we have these typical rings, and then the KR-like domain are underneath and linking those structurally together. Now, next we were wondering what the role of this additional domain might be. So we already could show it plays a structural role, but we were wondering if there isn't more to the story. So I mentioned before that this is a homolog to this KR methyltransferase, and the substrate for this methyltransferase is called SAM. So SAM is actually widely used in all domains of life. So it's a very common co-substrate involved in methyl transfer group as methyl transfer groups. It's a regulator of of DNA or RNA methylation. It plays a role in immune responses, amino acid metabolism, and actually it's an FDA-approved medication to treat depression, some forms of pain, and cancer. So we were wondering why there would be a SAM binding enzyme coupled to these receptor arrays. Now, unfortunately, we could not crystallize this domain, but we could use AlphaFold to predict the structure. And it's actually very similar to the methyltransferase. It also has the SAM binding pocket, which you can see here. But so as I mentioned, it can't methylate the receptors. Now, next, we worked with our collaborators, Chris Lee in Virginia Tech, to determine what the role might be. So actually, we did a very simple chemotaxis assay, which is a very classical experiment in the field. So what you do is you basically fill a capillary with a substrate you want to investigate, and you stick it in a culture of your bacteria. And if there's the bacteria are attracted to the substance in the capillary, they swim up the capillary, and the ratio between the cells outside and inside the capillary changes. So if they're attracted, it will be above 1. If it's a toxin and they can sense it, then it will be below 1. So what you can see here, that they're actually attracted to SAM. And if you delete this compound, then this chemotaxis goes away. So this domain enables chemotaxis towards SAM. And so we also did a control that actually does not affect chemotaxis towards other substances. Like, for example, in hemin is a known attractant for treponema. So removal of this domain doesn't affect chemotaxis behavior there. So it's really a specific SAM sensor. Which we find kind of exciting because I think it's the first described sensor of the chemotaxis system that bypasses the chemoreceptors themselves. So I think I've showed you so far that our work is really quite visual. So we decided that this project specifically, we wanted to actually bring that to the general public. And this year, Anthony van Leeuwenhoek has been dead for 300 years, so that the Netherlands was celebrating the life and accomplishment of Anthony van Leeuwenhoek, who was born and lived not far away from Leiden. It's a 15-minute train ride. So our local science museum did an exhibition on him for a whole year. So my student and we worked on generating a movie that the public would understand a bit more of our research. So this is a snippet of the movie that we made for this museum. So if you manage to come by Leiden, you should stop by and watch the whole thing. And this is the opening day, and Vera, who worked on this project, is very proud to see her work on exhibit in the museum. Now, when I first moved to the Netherlands, and I wrote my very first research grant here to get money to do this type of research, one of it was denied, and the explanation was it's such fundamental research and it doesn't have really applied impact. So we decided, let's see if we can actually turn this into something that we can use. And we decided to see if we can build basically a biosensor for a cancer called neuroblastoma. So that's mainly a cancer that afflicts little kids. So we decided to use the most basic system we have, so the E. coli cells, and see if we can build a biosensor. And so at the time we started, we thought, well, the final goal should be something like a pregnancy test. I think nowadays everybody's more familiar with the corona test. So that was the idea, to see if we can use E. coli to build such a cheap and fast neuroblastoma cancer screen. So why did we decide to go for neuroblastoma and use E. coli? So there's a very simple reason. So for in neuroblastoma, it is also already detected by substances in the urine of the patients. And these are called DHMA and VMA. And you can see here in healthy patients, the concentrations of these substances is very low. It's only 1 to 2 micromolar, but in patients, in the case of DHMA, is 50 micromolar, and in VMA it's even 1 to 3 millimolar. And DHMA was already a known attractant for one of the chemoreceptors found in E. coli. And VMA is structurally very similar to DHMA, so we tested that, and we could also show that it's also a chemoattractant like DHMA. And again, Keith Cassidy did some simulations to show how these compounds bind this chemoreceptor in E. coli. Now, first we did some very, very traditional chemotaxis assays to test this. So here, these are plug and pond assays. So it's very simple. You mix your compound in agarose pads, you put it in a light microscope, and then you can see if your cells are fluorescently labeled, that they accumulate at the border of this agarose pad if they're attracted. So here, in case of serine, which is a known compound sensed by this receptor, and then DHMA and also VMA, we could show the same type of attraction. And so here in a negative control, you can see there is no accumulation of fluorescent bacteria at this border. We of course wanted to do this more high throughput, so we make use of these microtiter plates. So these are basically 96 well plates with this channel system. So you have 2 channels, and one you can fill with E. coli and one with the attractant to test. And so the idea is that we have a high throughput screen to test these compounds and if the cells are attracted to it or not. So here are movies of these compounds with just mock samples. So again, VMA, DHMA, and also serine. It's very hard to see on my screen, but there is some accumulation here. Here with DHMA and VMA, it's very clear. So of course, so this is all unpublished. This is still work in progress. But we partnered with the Princess Maxima Centrum, which is a cancer hospital. So we started screening actually patient samples, and here are some of the results. So this is a healthy patient that doesn't have neuroblastoma. This is also a healthy patient with high serine in the urine. Just we wanted to see how much presence of serine might influence the output or the outcome of the test. And then we have 2 patients that I show here. So the first results are very promising. I think we have some more work to do to get this more consistent, but I think in principle, I think this might be a viable way forward to actually make use of chemotaxis in E. coli. Now I want to switch gears and talk about non-motile bacteria. And so this, I started thinking about this when I moved here, and like for 20 years I had worked on motile bacteria and how they sense their environment and swim around. And then I came to Leiden, and the whole microbiology department works on this non-motile species Streptomyces. So there are also very interesting organisms. They're main producer of antibiotics in the clinic use today, but they cannot move. So we were wondering, how do they actually get where they want to grow? And so here is just an overview of over the life cycle. So they start off as spores. These spores germinate. They form these substrate mycelia, so they're multicellular mycelial meshworks, and then they start at some point forming aerial hyphae that then turn into these spore chains that then can be dispersed. Now the current idea is that when the insects distribute these spores over wide distances, But how do they get to the final position where they start germinating? And they like to grow at the roots of plants. So how do they get there? And so this project was taken on by an outstanding postdoc in the lab, Dr. Elise Mork. She's also an artist, so she can draw her science in an amazing way. So how do the spores from the streptomyces here get to the root of the plant where they like to grow? And so Elise started doing some very, again, classical experiments in the lab. And so what she did, so she mixed a motile cell bacterium, bacillus, together with streptomyces spores and spotted it to the center of a growth plate. So you can see here in A, and then she just let it grow. And then the white spots you see here, these are germinating streptomyces spores. So in the mixture, the spores are distributed all over this growth plate. Now, if you just spot the streptomyces alone in the center, they just germinate where you put them. So just they stay in the center of the plate. So this is bacillus alone. Of course, they can cover the whole plate. And then she did, I don't know, hundreds of plates where she did experiments like this. So here in black, with the black asterisk, is always where she put the bacillus strain. And then here you can see the streptomyces. And so the streptomyces spores are always distributed over the plate in the direction of the moving wavefront of the growing bacillus cells. So here we have two. Again, it goes into the direction of the growing bacillus cells here. And here it's again, always in the direction where bacillus moves. And here, we are not plant biologists, but we managed to grow an Arabidopsis here on the plate. So here is the root of the plant. And again, you can see when in the condition where we mix the streptomyces with the motile cells, the spores can germinate where the roots of the plants are. Whereas if you spot it in the middle here and the plant is on the side here without the motile cell bacterium, the spores cannot reach the plant. Now, of course, we were microscopists, so we wanted to see what's going on here. And here is a low magnification image in the microscope. So here these long rods are the bacillus cell, so the motile cell bacterium. And then you can see the spores. And the spores seem to be always close to the pole of the bacillus cells, but never really touching. So we already had the suspicion here that the spores attached to the flagella of the motile bacterium. And so this is what we could also show here with light microscopy. So we sheared off fluorescent flagella. We could mix it with the spores, and you can see the flagella really wrap around and attach to the spores. So then we did some tests with mutant strains of bacillus. So we, with the outcome that the presence of the flagella was absolutely required for this transport, but actually active rotation of the flagella was not. So if the bacillus cells could still slide on the surface, the spores could still be transported. So here are some slices through a tomogram. So this is a streptomyces spore. So these spores are very big. So they're borderline for our technique tomography. So anything thicker than that, you wouldn't see much inside. But I think you can still see the thick spore coat of these streptomyces spores here. And so the spores have an additional proteinaceous layer outside called the rodlet layer. So it's like a sleeve and encasing the spores. And up until now, no one really understood the function of these rodlet layers. With SEM, if you look from the top here, you can see that these rodlet layers form these structured patterns on the spore surface. And intriguingly, oftentimes it looks like a train track with a distance from each other, from the two rod layer, from two rodlet protein tracks of about 20 nanometers, which is intriguingly the width of a bacterial flagellum. We also looked into the other part on the spore side. So is this rodlet layer really required for this transport? So we tested different Streptomyces strains that you can see here. These ones here, the first four, are all Streptomyces species that have such a rodlet layer, and they're all transported via this rodlet layer, and then they attach to the flagella of the Bacillus species. There's one here that naturally lacks the spore coat, and they're also barely transported. And if we genetically remove the spore coat from our model strain, Streptomyces silicolor, they're also not transported anymore. So we need the flagella and we need the spore coat for this transport to occur. So here is a tomogram that shows this again. So here we go through the spore, and then we have a segmentation of the spore body. In purple, the spore coat, and in yellow, the flagella. So at the moment, we're trying to shear off the flagella and mix it with the rodlet layer that are also removed from the spores to get a better idea on the more intricate interactions between the flagella and the spore coat, how this transport is facilitated. Now, lastly, I want to switch to host infection of bacteria inside a host. And so this is a very difficult system to work with when you think about tomography, because we're very limited with the size. I mentioned that before with the spore, so that the thickest we can image well is if our samples is 500 nanometers or less in thickness. The spores were about 1 micrometer, so really, really large. So if you think about going into a host tissue, it has all kinds of challenges. So we need to figure out how to make our samples thin enough to look through. But of course, that's what we want to understand, right? To understand the interaction between a host and its microbes. So I think that it's going to be like really powerful if we can start studying these systems in such a way that I showed you before. So the system we decided to first work on is what you're seeing here. So it's the Hawaiian bobtail squid. So why on earth would we start with this? The reason for this is quite simple. So these squid, they have a light organ inside where they accumulate one species of bacteria inside that then have a luminescence. So these light organs help them glow in the night and help them hide them from predators from below. So if these squids are only like this big, so if they're very tiny, and so if they swim around and they can emit this light, predators from below can't really see them against the moonlight from above. And so this is, I think, the most simplified system between a host and a microbe, because it's one microbe and one host interacting with each other. In that sense, it's a very simplified system that allows you to really start studying such an interaction from a more simple way. Because if you think about humans, you have thousands and thousands of species, and it's really hard to disentangle who is who and who does what. So this is a nice system to start with. However, of course, you can see that these are of course, much bigger than 1 micrometer. So here you can see us actually how we collect them. So this is the adults are all collected in the wild. So there's one beach in Oahu, Hawaii, where you go out after dark in knee-deep water with a flashlight here, my postdoc and myself. So you catch these, and then they're grown in a few labs over the US, in Hawaii and over the US, and bred. And then the eggs that they produce, they can be shipped to the Netherlands, where we have now our squid lab. And this is, I think, one of our first, oops, let's see if I can play this again. There we go. This is one of our first hatchlings that we could raise in our own lab. So here, you can see again one of the juveniles. So the juveniles are really quite tiny. Here, this dotted box here shows the location of the light organ. It's right on top of the ink sac of the animal. Here's an SEM image of this light organ from this juvenile. So it has these appendages here that have these cilia on top, and so the cilia move around and move the seawater, and basically guide marine bacteria to the openings of this light organ. So a lot of bacteria of this similar size as Vibrio fischeri, which is the cell that are taken up, are accumulated here, but then only this one species can actually enter into the crypts of the light organ that are shown here in this cartoon. And so this has been studied for about 30 to 40 years. And so a lot of beautiful light microscopy has been done, but we really want to understand from a structural level, how do the bacteria attach, how do they use their appendages and molecular machines to gain access to these light organs, and what makes that so specific to this one species. Now, one of the molecular machines that is implicated in this interaction with the host and the specificity of these Vibrio fischeri cells going into this light organ are type 6 secretion systems. So the species has two. There is beautiful work by many labs done on different type 6 secretion systems in different species. So here's one where you can see here the type 6 secretion system are these long needle-like structures with this membrane component here. So it is known to help species in microbial combats, for example, when they can battle other species and kill them using these structures, but they're also implicated in intricate interaction with the host. So here, for example, here's one that was done from the Grant Jensen lab in Myxococcus, and none of these systems that are so far described have really prominent extracellular features, except in this one, there are these filaments that were seen by our former colleague Yi-Wei Chang in his beautiful paper here. So when we grow Vibrio fischeri now in viscous medium, so that's not in the squid, we just grow them in a mucus-like environment, and we can look at their type 6 secretion systems. We find they have these pronounced large extracellular structure outside. And so my postdoc who works on this, Katrina Gundlach, she thinks it looks like a beer bottle. This is the brewery where her husband works, so maybe that looks like it. So we hope to see in the host if this extracellular feature is really involved in the interaction with the host cells. Unfortunately, this is not very regularly ordered, so we can't really do subvolume averaging on this. So this is for now the best we can do in terms of resolution. But now our real goal is to understand how this really looks like in the host. So here So this is not cryo yet. So this is work done by Mark Ladinsky at Caltech, who collaborates with us on this. And so we do plastic embedded samples and huge montages of bacteria in context with the squid, and we can see that they're tightly embedded. So here we can see the crypt spaces. It's much more complicated than we originally thought from just looking at the light microscopy. Here, these dark dots here, these are all bacteria, but these crypts are very narrow layers. So there's basically only one cell layer thick. So only one bacterium basically fits into some of these spaces. And you can see they're in very close contact with the microvilli of this inside the slide organ. But this plastic embedded sample is not good enough to really let us see the structural features like the type 6 secretion systems or the flagella or pili or the chemosensory systems of these bacteria. So we really need the next step of cryotomography to look into this. So for this, we had in mind a very elaborate working protocol from vitrifying the samples, pre-trimming the huge samples with a cryo-ultramicrotome, correlative microscopy, and then using FIP milling, which I think is really a key technology for the coming years. This is a scanning EM with an additional ion beam inside that you can use on very thick samples to mill away excess material to basically cut windows in your thick samples that then can be used for cryotomography. Now, the challenge that we currently have is shown here. So this FIP milling is very powerful, and people have used it for amazing insights already. And I think already, like HeLa cells are already considered large volumes, but I think this is already now a standard technology looking into HeLa cells and milling those. So this is what currently is the standard of large volume. The current record in size that people have done is a C. elegans worm. I think it's still on bioRxiv, but it will come out in Nature Methods soon. So this is done by the Max Planck Institute in Martinsried, by Jürgen Plitzko, and the PhD student Oda Schütz that has done amazing work. So in comparison, this is to scale the size of our light organ compared to the C. elegans worm. So it's another challenge. So we approached the Jürgen Plitzko and Oda Schütz to see if they would be interested in helping us figure out how to process the light organ, and luckily they were interested. So we're collaborating with them now. So here you can see a light organ glued onto an EM grid. And then my postdoc, Katrina, went to the Max Planck Institute to work with Oda. And so what Oda does, so she developed this serial lift out technique. So what she does, she uses this ion beam to cut out this block of ice here in this green striped color. And then she puts it on the EM grid and she slices off the bottom layer. So she has a thin layer of sample on the grid. And then she goes to the next position and slices off the next piece of the block. So she can serially transfer the block of sample and put it in slices on an EM grid. So this was our first attempt with her. So we had here 12 lamella. This didn't result in data collection, but we went again. And now we have a first set of about 40 tomograms, which is super exciting. It's just a couple weeks ago, so this is very preliminary. We just missed the bacteria, so I think the issue here was that this was from a squid that had just vented. So these squid, they every morning they kick out 95% of their bacteria. They just push it out into the ocean water, and they just retain about 5% of the population that then grows up again overnight. So this animal had just vented, but we could get amazing tomograms of the squid tissue inside of the slide organ. And here's just a couple examples. So one, what you can see here, this tomogram that's playing, you can see, I don't know if you can see my mouse. You can see a nuclear pore. And so for those of you who know nuclear pores, they're quite well studied. There's beautiful work, structural work done. There was always this mystery that in the squid, it is known that these nuclear pores are leaky. So things can enter into the nuclei that is not possible in for example, human cells. And looking at these tomograms, we exactly know why, because these nuclear pores are much wider than they are in other organisms, and there is less structure inside. And then we could also start to do the first subvolume averaging of some of the proteins found inside these tissues. So this is super new. We just started working with these tomograms, but I think it's very exciting to see, because it is possible to do tomography of samples of that size. And I think that opens up a whole new world for biology. I'm very excited to see where our lab and also many others are going with this, and what kind of biology we can address in the near future. Oh, sorry. So I think I'm out of time, right? So I just want to go to the acknowledgements. So this is work done by my amazing lab. So the work on the treponema cells and the spore transport was all done by my postdoc, Alice Muak. Vera was the one who did our animations of treponema and made the movie for the museum. And Adam did the experiments with the neuroblastoma sensor. And Katrina is the marine biologist that does the Vibrio squid work. And so I hope next time I can tell you what all these other amazing people are doing. And of course, we have fantastic collaborators. Without, we couldn't do anything. So Margaret McFall-Nagai and Ned Ruby are our squid collaborators that are the main people in the squid field. Mark Ladinsky did the traditional EM for us, and Oda and Jürgen the large volume, and many others. And of course, nothing we can do without our funding. So I'm very thankful for that, and I'm happy to take questions.
Thank you so much for delivering a great talk. So, yeah, are there any questions from the audience?
I can ask a question quickly.
Yeah.
Hi, Ariane. Great to see you. Great to see chemoreceptor arrays again. No, it was fantastic. A really wide variety of projects, and really all of them are very, very exciting projects. So I have a couple of questions. The first one is related to chemoreceptor arrays. So just the way in flagellar motors, for example, where the bacteria is living, depending on that, on the viscosity of that medium or surrounding, flagellar motors will have different strength. The stator complex can be more sort of complex. Do you see compositional diversity of chemoreceptor arrays based on the environment of the bacteria?
Yes. So yes, absolutely. And I think it's we're just starting to look into the differences. And I think maybe one example is Vibrio cholera, where it's really dependent on the environment. So as a bacteria that switch environments traditionally have many more receptors. Like E. coli has 5, Vibrio has about 43. And they turn over, so they switch out depending on which environment they're in. So we know that also from proteomic studies that they change. But structurally, so in E. coli, these arrays, I showed you this super ordered architecture also with the kinase underneath. In Vibrio, the stoichiometry is very different. So there's much less of the kinase in the array, and that actually makes them a lot less stable. So we think this is actually an adjustment to the environment, because it allows the cells to turn over the receptors. So they can actually adjust the receptors that they need depending on the environment. Whereas in coli, it's once the receptors are embedded in the array, that's where they stay. They never turn over. It's very, very rigid. But in Vibrio, it's not. So, but I think we have to look at many more bacteria to really, see how different organisms really do this. So we can only tell currently from Vibrio, Treponema, and E. coli. And we recently started working actually with arrays of the Loki archaea that were given to us by Martin Pilhofer, our good friend. So he found chemoreceptors which he donated to us. So we're analyzing those, and they are again differently arranged. So it's very exciting.
I have another second question, and this is regarding the rodlet structure that you are showing on top of the spores. Do you have any idea like what those could be? Any idea what exactly they might be?
So we know, so we know the proteins. So they're different. They're called rodlet proteins, or rodlet A and B, that form these structures. So we know the composition of it. But we don't have high resolution of this yet. So we need to strip off the
slate. Are they secreted?
Sorry?
Are they secreted? Are they secreted?
Yeah. Yeah.
Okay. Thank you.
By the way, before I forget, whoever is interested in tomography, you guys are going to get the next tomography congress in Australia. I think it will be spring 2025. So early, about a year from now. So.
Okay. Georg?
Yeah, thanks, Ariane. Beautiful talk. I just had a question about the last part with the MP and the serial imaging, and the specimen you showed, it looked like it was plunge frozen, or was it high pressure frozen?
Oh, that's because high pressure.
It was on. It was high pressure frozen, but then in the autoloader assembly.
Yeah. So what we've done is we actually Katrina, the marine microbiologist, so she cuts out the light organ, and then so in that experiment we glued it on an EM grid, and then we put it into the high pressure freezer. That might be unnecessarily complicated, so I think actually we're gonna skip the gluing on the grid because it's not necessary. But for historical reasons, this was our approach initially. But yeah, it is high pressure. Okay. So I think so, because these light organs are actually quite huge, right? They're 200 micrometer in height and 700 micrometers in width. So they're quite big.
Cool. And there were some black dots which look like gold fiducial. Is it some internal electron and structures?
No, that's redeposition of the coating from the yeah. I asked the same question. It looks like gold beads because it's also super
exactly
inside. Yeah, no, it's actually luckily just redeposition of the coating. That makes actually reconstruction quite easy.
Yeah. Okay. Thanks.
Thank you. I have a quick question in regarding to the biosensor work. So how sensitive are those?
So we haven't tested the exact range in our high throughput system. However, the coli chemotaxis system, it's very sensitive.